Clearly, blood tests are important. Have you ever asked what exactly blood tests measure and how they do this? Let's take a look at how blood tests work. The basic principle of immunological-based blood tests lies in our ability to measure the presence of pathogen-specific antibodies. When B cells encounter a pathogen, they produce antibodies exquisitely specific for the viral antigen in question. Thus, when a person is tested for HIV, for example, we in essence test whether or not that person produces antibodies specific for HIV antigens. The presence of such antibodies is telltale proof that B cells have encountered the virus, undergone clonal expansion, and now secrete thousands of IgG molecules into the bloodstream. Thus, the task becomes how to measure HIV-specific antibodies. This is done using a laboratory method called ELISA, that is, enzyme-linked immunosorbent assay. Let me walk you through a typical ELISA blood test. I've asked Michael to join me and assist me. First, we apply proteins to the wells of a 96-well plate. The proteins simply stick to the plastic. The following scheme shows the general layout of the experiment we are about to conduct. To the columns, we will apply antigens from HIV, HCV, hepatitis C, and 3 flu strains, H1, H3, and influenza B. For each virus, we apply duplicate wells. We leave the last 2 columns without any antigen as negative controls. Applying duplicate samples will give us confidence that our results are credible and can be faithfully repeated. Here, Michael applies HIV antigens to the first 2 columns of the plate and proceeds to columns 3 and 4 with HCV, and so forth until the entire plate has been loaded with samples. The plate is incubated overnight, and in the morning, excess unbound proteins are washed away. Then we add a blocking solution containing milk that blocks any plastic surface of the wells that still might be able to bind proteins and reagents to be used in the coming steps of the test. This blocking solution helps reduce nonspecific sticking and background signals. After incubating the plate, the blocking solution is discarded and we wash the wells again. Now, the blood samples from the people being tested are added. The samples are serum samples, that is, blood samples from which we have removed all the red and white blood cells and use only the remaining fluid of the blood that contains antibodies. In this experiment, we have taken serum samples from HIV and HCV infected individuals. For flu, we apply standard sera of flu-inoculated sheep. Row F contains buffer without serum, and G is serum taken from a healthy volunteer, me. Thus, rows F and G should provide negative controls. Here, Michael applies the serum of the HIV-infected individual. Each well receives the relevant serum sample and is incubated for an hour. Afterwards, the wells are washed to remove excess unbound antibodies. Then, we add to all the wells enzyme-linked detectors. These are reagents that recognize and bind human and sheep antibodies and have also been chemically linked to an enzyme that can convert a chemical dye from being colorless to bright blue. So, if, for example, the HIV-infected person has antibodies directed to the HIV antigen, these antibodies will exist in the HIV wells and be able to bind the enzyme-linked detector. Similarly, HCV sera will react with HCV antigens, flu for flu, and negative controls should remain negative. So the plate is washed. In order to reveal the presence of the antibody-antigen complexes, all we now need to do is add the substrates for the enzymes linked to the detectors and see where the dye changes color. As you can see, some of the wells developed a strong blue color, indicating a positive result. Good job. Careful examination of our results illustrates that color develops in the serum samples as to be expected. HIV-infected people have antibodies specific for HIV antigens, but do not develop signals for HCV. HCV sera react with HCV antigens as expected, but not for HIV, and so on. The flu-inoculated sheep respond to their corresponding flu viruses, with some degree of cross-reactivity with the other flu strains. And I'm clearly negative for HIV, HCV, and, wait, it turns out that I'm positive for all 3 flu viruses. I feel great, I'm healthy, and I don't even have sniffles. Does this make sense? Actually, yes. You see, I've been vaccinated, and as we will see in the coming lesson, vaccination allows us to produce virus-specific antibodies without having to experience the disease. Vaccination is a good way to protect ourselves from infection and keep us healthy.